Thanks very much, Patricia. It's a great pleasure to be here, and thanks everyone for staying to the bitter end to hear me speak. I appreciate it. So you could make a pretty good case for the mammalian retina to be in the best understood element of the central nervous system, and yet it's only just less than 15 years now since we knew that it can work without rod and cone photoreceptors generating visual information thanks to the presence of a new photoreceptor here in the inner retina. And so what I wanted to do today was to give you a brief overview of the discovery of that photoreceptor and its form and function. And unfortunately, I'm going to have to race through that, so I won't be covering important work from colleagues around the world, but just to give you a flavor of how it panned out. And then I want to spend the second half of the talk concentrating on how already in small ways it's starting to change the world around us and how that might evolve in future. So why the discovery of these photoreceptors could end up being important for our everyday life. Okay. So the story starts with my own interest at the time, as Patricia said, which was circadian rhythms. So we've heard a series of nice talks about circadian rhythms here, and I'm sure we're all aware that we have a biological clock in our hypothalamus, in the suprachiasmatic nuclei here above the optic chiasm, in which neurons generate a rhythm with a period of about 24 hours. And that rhythmicity is imposed then on practically every body system. So we're very well aware of our sleep-wake cycle, but almost every biological system will have a 24-hour variability, a 24-hour rhythm. As I say, this rhythm is generated by an endogenous clock. But of course, its adaptive significance is allowing us to predict periodic changes in our environment. And that's only possible if that endogenous clock is reset to local time by some environmental signal of time of day, okay? And we're very well aware from our own everyday experience that by far the most reliable environmental signal of time of day is the light-dark cycle. So ambient light can be 10 billion times higher in the day than at night. So it's a big signal. But also, importantly, there are very systematic and predictable changes across the dawn and dusk transition that organisms can use to pretty accurately tell time, at least around those times. So one might expect then that this signal is what circadian clocks use to set to local time. And indeed, across the living world, that turns out to be true. And we can see that in the lab by studying the organisms that we work with, laboratory mice, very nicely. So this is an activity record from a single lab mouse in which all we do is we house the mouse in a cage where we can monitor revolutions of its running wheel, okay? And then we plot what's called an actogram, where every horizontal line here is 24 hours, and every time the wheel rotates, we get a little blip. So the more black you get, the more that mouse is running in its wheel. Okay, so we have, this record shows about 10 days of recording, where we go from all the way through here to midnight, and then we start again on the line below. And I've shown above here clock time, and the time at which lights in our vivarium go on and off. Mice, at least in the lab, are nocturnal animals, and you can see that this mouse is very reliably restricting its wheel running activity to when the lights are off. Okay, so it starts out with a pretty strong indication that light is telling this animal when to be active. To test that, we can shift the light, the equivalent of taking our mice from Manchester to Perth in Western Australia. And what happens is that over several days, the time at which they get active moves forward. So this is the mice having jet lag, and then they resynchronize to the new light-dark cycle. And you can shift them back, and gradually they go back. And at this day, we can put them in constant darkness, and you can see the rhythm here with a period of roughly slightly shorter than 24 hours, meaning that the mouse gets up slightly earlier every day in constant darkness. Okay, so what this record really shows is that, leaving any other influence aside, if we change the light-dark cycle, the timing of light exposure, that defines the phase of the clock. Okay. And as I say, that's true across the living world. So, where does that leave us? That tells us that the 24-hour variation in ambient illumination is a very important information for the clock because it tells us what time of day it is. And so practically all organisms on Earth require a mechanism for tracking it. Okay. So if we think about a very simple organism like this Neurospora, it has to have a light sensing mechanism there which is measuring the amount of light in the environment and resetting its clock, and indeed they do. Okay. So what about a more complex organism, and particularly an organism that also has a very highly developed visual system like this bird? Okay? Vision also relies on photoreception, on measuring light. So one might ask how compatible those two reasons for measuring light are. On the one hand, setting the timing of our biological clocks, and on the other hand, supporting vision. And there's an important reason why they're pretty incompatible at some level. And that is that 24-hour variation in ambient light is a strong challenge to the visual system. So if we put ourselves in the position of this chap reading his newspaper in the Dead Sea here, bright, bright sunny daytime, compared to this Japanese lady reading a poem by moonlight, under both circumstances, their visual systems are trying to measure small differences in the light reflected from objects in their visual scene, and yet the total amount of light between these can be many, many orders of magnitude different. And so, it won't come as a surprise to anyone here, just to hear that visual sensitivity is adjusted according to ambient illumination. Okay? So our photoreceptors and the networks downstream from them are adjusting their sensitivity all of the time to try and limit the impact of this big variation in ambient illumination. But the process of doing that means that to some extent at least, you're deleting the information about changing background light intensity. Okay? So although both of those functions require fundamentally photoreception, you can see that at some level they're fairly incompatible, at least to some degree. So that's a conceptual argument. Do we have evidence from the living world that this actually is the case? And we do, because if you look at non-mammalian vertebrates, fish, amphibia, birds, and reptiles, they separate those two photoreceptive mechanisms, okay, in anatomical terms. So this is a lizard, and we're looking at the top of its head, and of course it has lateral eyes here that it uses for seeing, but it also has this organ here, which is just beneath the skull, and you can see this kind of thinning of the skin here, and that's its parietal eye. It also has photoreceptive neurons deep within its brain, has a photosensitive pineal gland. Other organisms have photosensitive parapineal organs. And we now know that many organs distributed across the animal, starting with the skin and moving down to the heart and the liver, are themselves directly photosensitive. So for these organisms, they have photoreceptors outside of the eyes that they can use to measure ambient light intensity. So you can have a very clear separation. And we've known about that for a long time, at least 50 years. Okay. So what about mammals then? And the paradox in mammals is that if you remove the eye from any mammalian species, not only does it not see, but also its biological clock can no longer track light-dark cycles. Okay? So that tells us that, unlike all other vertebrates, for whatever reason, we restrict our photoreception to this organ, the retina. Okay. So how then does the retina deal with this dichotomy, the varying demands of measuring ambient light compared to supporting form vision? And that was really the question that the first experiment that I did in this field addressed. And in that, we asked whether, if you genetically remove rod and cone photoreceptors from the retina, you can still get those circadian light responses. And the answer is that you can. So this is an activity record from one of those rodless coneless mice, and it's actually the same activity record I showed you before, showing that this animal is very nicely tracking the light-dark cycle despite the fact that it lacks rods and cones. Okay. So that tells us then that somewhere here in these neurons in the retina, there must be a way of directly measuring light. Okay. So this inner retina doesn't only rely on rods and cones for visual information, it can generate it by itself. Okay. So what are those photoreceptors? And the really seminal discovery in this field came from David Burson's lab at Brown University, and he was recording from these cells here, retinal ganglion cells, the small fraction of retinal ganglion cells that project to the suprachiasmatic nucleus and therefore convey visual information to the clock. And what he discovered was that if you isolate those retinal ganglion cells from synaptic input, they still respond to light. And actually, this wasn't from his first paper, but one of his subsequent papers. These are current clamp recordings of those retinal ganglion cells in ACSF and synaptic block conditions. And what you can see is that when you turn the lights on here, you get these cells are excited by light and they generate lots of action potentials throughout the time the lights are on. And that also happens when you have your synaptic block. But if you look closely, when you have the synaptic block, you get this nice maintained firing, but you lack the very immediate responses to light. Okay, so what does that tell us? That there are a bunch of retinal ganglion cells that are excited by light even when isolated from rods and cones. And he called them intrinsically photosensitive retinal ganglion cells, or IPRGCs. Kind of wish he'd come up with something snappier, but there you go. The IPRGCs have their own intrinsic light response here, but because they're retinal ganglion cells, they're also connected via these neurons to the rods and cones, and so they merge these two information streams, and you can see that you can see the effect of that here, where thanks to their intrinsic response, they have this nice maintained activity, but they use their extrinsic inputs to have very sharp and abrupt responses to changes in light intensity. So the influences differ in the time domain, with the intrinsic response being slow but sustained. And since then, we and others have found them also in mice and in primates. So they exist across the animal, across the mammals. Okay. They're a tiny fraction of ganglion cell population, 1 to 5% depending on the species. So how do they absorb light? How is it that they become to be photoreceptors when all of the other retinal ganglion cells aren't? And the answer is that they express a protein which was first discovered in the skin of this species, Xenopus laevis. And the Xenopus skin is photosensitive, and you get changes in pigment aggregation depending on light exposure, and that relies on a protein called Melanopsin. And that was a discovery from Iggy Provinciaux and Mark Rolag with their uniform services, that there's this protein Melanopsin which provides that function. And it turns out that Melanopsin is expressed in these few retinal ganglion cells. So Melanopsin is related to the rod and cone opsins that our rod and cone photoreceptors use, but perhaps even more slightly closely phylogenetically related to the opsins found in invertebrate photoreceptors. So it's been around a long time in evolution. Okay, so what else do we know about IPRGCs and Melanopsin? One of the most important things we might want to know is what kind of wavelengths of light they respond to. And that's been addressed in a few ways. And I just want to talk about one way in which we can do that, where we can make easy comparisons across species. And that is that you can express Melanopsin in HEC293 cells very nicely. And if you include a luminescent reporter of calcium, then when you flash those cells with light, you get a nice calcium response here in response to that stimulus. And when you look at cells without any opsin, then you don't get the same event. Okay, so we can make HEC cells photoreceptors simply by expressing Melanopsin. And that's very useful because then you can look at Melanopsins from different species and ask about their signaling characteristics and those sorts of things. I want to concentrate on one thing that you can do, and that is that you can change the wavelength of this stimulus and measure the response to look at the spectral sensitivity for Melanopsin. Hopefully this is going to come up. And when you look at human melanopsin in these cells, which of course would be very hard to measure any other way, then what you find is that it has peak sensitivity in this kind of cyan region of the spectrum here, between 450 and 500 nanometers. It's actually most sensitive at 480 nanometers. And it turns out that if you put melanopsin from Xenopus, it looks like that. From chickens, it looks like that. From mice, it looks like that. So it's a very evolutionarily conserved feature of melanopsin photoreceptors, that they're sensitive maximally to this blue bit of the spectrum. And we're going to return to that later. Okay. What do IPRGCs look like anatomically? And here I've just shown some data from our own lab where we have been trying to use Brainbow technology to reconstruct the projection pattern and the anatomy of individual IPRGCs. So what we do is we take a melanopsin Cre recombinase mouse and we inject 2 viruses, each of which expresses 2 fluorescent proteins, whose expression is under control of the Cre recombinase at relatively low titer. And you get stochastic expression of these transgenes. So that you label individual cells here. So here's the optic nerve head here, and you can see lots of ganglion cells that are fluorescent. But depending on what the combination of the different fluorescent proteins they express, you can get a variety of different colors for these neurons. And so what you can see is that they're relatively sparsely distributed. I said there's relatively few of them. And then if you look at an individual, you can reconstruct the projection pattern, the dendritic tree of an individual cell by using principal component analysis to look at the relative expression of the different fluorescent proteins. So if we look at this cell here, which hasn't projected very well, unfortunately, then you can look at it when you reconstruct it and separate out all of its dendrites from its neighbors. And what we find is that actually there are 6 anatomically distinct classes of IPRGC, so you can tell already that there's scope for quite a lot of functional diversity. The other thing you can do is you can track then their projections in the brain, and this just shows a sagittal section through the dorsal LGN, and you can see even from a relatively sparse labeling of these ganglion cells, there's lots and lots of axons going through the LGN. And again, here you can look at an individual ganglion cell's projection, and what you see in all of the brain regions we've looked at, that there's a lot of dendritic branching in all of the target areas. So what we find then is they project not just to the SCN, but to a variety of other nuclei driving visual reflexes, and also to the primary visual thalamus. So the DLGN is the retinal target from which thalamocortical projection neurons go to the primary visual cortex. And they have extensive axonal branching, which means that a small fraction of retinal ganglion cells can influence large populations of neurons in retinorecipient regions. Okay. I want to turn to another question one might ask, which is what is the function of these IPRGCs? So there's a lot of circumstantial evidence that they're involved in setting the clock. Is there any way we can directly ask whether that's the case? And so the route that we have used recently for that is to use this chemogenetic approach, which is available from Brian Roth's lab. And so what the Roth lab did was to mutate the muscarinic receptor to make it insensitive to its natural ligand, but sensitive to a synthetic compound, CNO, which otherwise is at least relatively biologically inert. And the idea then is that if you can genetically target expression of this receptor to a particular population of interest, and then apply CNO, then in this case you get activation of a G-alpha Q signaling cascade, which results in neuronal depolarization. Okay. So we've done that again using the Melanopsin Cre mouse to restrict the dread to IPRGCs. And so what happens then if you apply CNO to those mice? And the first thing that you can do is have mice on running wheels here. So you can see this is the light-dark cycle. The mice are entrained to that. And then in constant darkness. And then on this day, we give them CNO in their drinking water. And you can see that although the mice are still in constant darkness, you get this big shift in the phase of the circadian clock, telling us that these guys, that simply activating IPRGCs alone is enough to influence the clock. So what else do you notice about applying CNO to these mice? They get a really, really strong pupil constriction. So this is something we're all aware of, the pupil light reflex. When light intensities goes up, you get a pupil constriction. This is a mouse in complete darkness, and its pupil, which should look like this, looks like that. There are also fundamental changes in behavioral state, so you can see that in open field tests. So in the open field test, when mice are anxious or alert, they tend to stay around the outside, the perimeter of an open field. And when you apply CNO to our mice expressing the dread, then they spend much less time in the center of the arena. And that's specific to the dread expressing mice, because if you do it to our control animals, then you don't see the same thing. And we also test that with an elevated plus maze. And again, anxious mice tend to stay in these closed arms of the plus maze compared to the open arms. And we see again that when we apply CNO to our dread mice, they spend much less time in the open arms of the maze. Okay. So among the things then that IPRGCs do, the selective activation of IPRGCs is to shift the phase of the circadian clock, constrict the pupil, and induce this alert/anxious behavioral state. So these very few retinal ganglion cells are having a very fundamental effect on this animal's physiology. Okay. So to summarize what I've told you so far, neurophysiological systems optimized for vision face challenges measuring background light intensity. And so if you look across the animal kingdom, there's been this separation between visual and circadian photoreceptors. But in mammals, it's all restricted to the eyes, but not just to rods and cones, because we have these IPRGCs, which are a small fraction of ganglion cells. They have poor spatiotemporal resolution, but drive that sustained response. They're directly photosensitive thanks to the expression of Melanopsin. And they project to the clock, and activating the IPRGCs is enough to reset the clock and also drive a number of other reflex responses to light. Okay. So the last point here is something that I want to develop for the rest of my presentation, and that is that these IPRGCs, the melanopsin pigment that they express, is maximally sensitive around 480 nanometers. Okay. So if we think about real-world applications of the discovery of IPRGCs, one of the things that we can consider is that knowing that there's a new way of measuring light makes us reconsider the artificial and architectural light that we are exposed to. So we can start with the fact that light at night is much brighter than it should be, and light during the day is much dimmer. We can also think about how blind people respond to light, and I'm happy to talk about that in questions. But what I want to talk about for the rest of the presentation is how it makes us reconsider the appropriate colors of light, and how by changing color, that can be used from a practical perspective and also as an experimental tool. So to introduce that, I just want to take you through one of the most important kind of developments in visual science in terms of practical application, which was in 1931, the CIE, which is the standards authority for light measurement, agreed this RGB color model based on really beautiful psychophysical experiments over the previous decade. And what that means, and the nature of that, is completely apparent once we look at the practical application of it. So consider a scene such as this, which is just a natural scene. If you view that scene when you're just out and about in the real world, then what you're actually responding to is a distribution of wavelengths of light across the whole visible range. Okay? So the light reflected from elements of the scene will have a very complex pattern with with wavelengths at every, with energy represented at every wavelength. Okay. So what the RGB color model does is appreciate that you can simplify that enormously by taking 3 images of the scene. One that's red, one that's green, one that's blue. And then you can recreate our perception of that scene by mixing light, a blue light, a green light, and a red light. And of course, these are matched to our 3-cone photoreceptors, red, green, and blue photoreceptors. So what we are looking at here, actually, for both of these projected images, is nothing like the light that's really produced in the real world. It's really a trick whereby we're mixing just 3 different types of light to recreate our sensation of what we see here. And that's, of course, a really, really powerful concept. Okay. So to see it in specific action, here is a banana. And if you measure the light reflected by the surface of the banana here under sunlight, it looks like this. And if you measure the light produced by a computer monitor reproducing a banana, it looks like this. And you can see that they're fundamentally different, physically very different lights, but look similar to us. Okay. So what that RGB color model does is it allows for artificial light in images not to recreate the range of wavelengths of light that are actually found in nature, but rather the human perception of them. Okay? And the assumption built into it is that this can be achieved by controlling the activity of cone photoreceptors. So you can see where I'm going. If you assume that in the daytime, under kind of brightest light, all vision originates with cones, then the RGB model works very well. But as soon as you have a new photoreceptor, let's say Melanopsin, then it's worth reconsidering how well this model works. Okay. And it extends not to visual displays only, but also to artificial and architectural light here. So, this hasn't projected very well, but this is where I live in this very, very dark part of the world, it turns out, which is Manchester. And very occasionally we have heavy cloud. And if you go out on a day with heavy cloud, you can measure the natural spectrum of light. Here's daylight in Manchester on a heavy cloud day. And here is what the designers of our fancy building decided constituted good light for my office. So this is the light measured in my office. And it's produced by a fluorescent light source. Okay. So again, you can see that this red line here, this light, really is in no way a reflection of daylight, true daylight. So why is it that this is considered acceptable? Again, it's because it has the right balance of shorter, medium, and long wavelength light for it to look white to me. So it's recreating the balance of cone photoreceptor activity of daylight, and that's why it looks white. Okay, so that's fine, except when we start thinking about Melanopsin. So here is Melanopsin's spectral sensitivity, peaking at 480 nanometers, and you can see that it falls in a part of the spectrum here where my artificial light has much, much less energy than daylight. Okay, so in effect, what the artificial light in my office does is recreate the color of daylight without its Melanopsin efficiency, effectiveness. Okay, so that's what happens with the white fluorescent source. But of course, there are now a huge array of ways in which you can generate light. And here's the emission from a white LED. And you can see that has much more output here in this blue part of the spectrum. So you can look at these two lights, both of which would be acceptable white lights for an office environment, and yet this is has a much stronger activation of Melanopsin than this one. So white light comes in different forms. You can have Melanopsin high and Melanopsin low versions, and there's scope to build this understanding into decisions about how much you want to influence your circadian clock, for example. You could change the light at daytime and during the night. And that started to find its way into architectural design. Okay. What about visual displays? Okay. So remember I said that visual displays have RG and B components. And so in this regard, there can only be one way in which you can make these colors. Okay. Unless you reconsider visual display design and add an additional color. Okay. So what you can do is replace your RGB with 4 different wavelengths, 4 different colors, a violet, a cyan, a yellow, and red, or V-cry as we call it. And then if you do that, then you can create the same color either by mixing lots of violet, yellow, and red, and a little bit of cyan, or by mixing lots of cyan and red and a little bit of violet and yellow. And you can produce images whose color and saturation look exactly the same, but that differ fundamentally in how much they're exciting melanopsin. So this is a melanopsin low version, and that's a melanopsin high version. So this concept can be extended also to visual display units. Okay. So implicit in what I've said up till now is that you can do this because melanopsin doesn't really impact vision. And it's really about circadian clocks and reflex responses. But I also told you there are a number of different types of IPRGC, and melanopsin is expressed in different types of retinal ganglion cell. And in fact, we know that melanopsin can, or there's pretty good evidence that melanopsin can contribute to our perceptual vision. And that's what I'm going to talk about now. So some of our IPRGCs go to the visual thalamus, which is the origin of thalamocortical vision in mice and primates. And so we in the lab have spent some time looking for evidence of melanopsin responses in neurons in the LGN. And the approach I'm going to show here is to use these silicon recording probes, multi-site recording probes, targeting the LGN in anesthetized mice. And we can present 2 sorts of stimuli in these experiments. One is a simple light step, so we just go from dim to bright light. And the other is this stimulus. So we start with a magenta sort of light, and then we put a cyan on top of it. Okay, so why do we do that? Well, that's what we see, is this change from magenta to cyan. But actually, for the mouse, this magenta represents melanopsin low, and the cyan melanopsin high. So that for the mouse melanopsin, what it sees is a flash like that, whereas for mouse rods and cones, what it sees is that, an invariant target. There's no change in luminance for rods and cones because of the combinations of wavelengths that we use to generate those stimuli. Okay, so what happens, and we call that the melanopsin step. So if you record activity in the LGN, you find a large number of neurons that do this. So in the red is their response to a light step, so this very sharp transient response here when the light goes on, and then sustained activity while the light's present, and then it dissipates. When we apply a melanopsin step, we lack this very sharp onset. It builds up gradually, but over the second part here, we've got almost as much activity from the melanopsin step as we do from the light step, which activates everything. So around 1/3 of DLGN neurons in the mouse respond to a stimulus which is only visible to melanopsin. So what is that? Do we have any evidence that that impacts their perceptual vision or their decision-making around their environment? So for that, we have used a trapezoid water maze, where we have asked mice to find an escape platform here based upon the presence of a light. So we train them, so for example, I won't show the movie because it's not really going to show properly, but you put a mouse in the back here, and it learns very quickly to swim towards the light to reach the escape platform. Okay, so how do we know whether melanopsin is contributing to that process, the assessment of brightness that those mice make? And we do that by using different colors of light for the experiment. So here we use a red light and a green light. Melanopsin is almost insensitive to the red light, so the green is very, very strongly brighter for melanopsin. Cones can detect both the red and the green, so whether they think the green is brighter depends on the intensity of the green light. So we start out by having the green light really bright, and we train the mice to go to that bright green light to find the escape platform. We can then remove the platform for probe tests, and what we do with those is we reduce the intensity of the green light. And so, when the intensity is moderately reduced, it's always going to be brighter for Melanopsin, the green, but you're going to get to a point that for cones, the green and red look indistinguishable, and then eventually the red light should look brighter. And just to remind you that mice lack red-green color discrimination. So this is a task they're doing only on the basis of how bright they see those lights. Okay. So what happens when you look at wild-type mice? When you reduce the radiance of the green target, when the green target's bright, the mice reliably swim towards it, and when it's dim, they reliably swim towards the red. Okay. Given that Melanopsin is always thinking the green is brighter, this tells us that wild-type mice can use cones to distinguish this brightness, the brightness of these targets. So no Nobel Prize for showing that. So why do we think that Melanopsin might be relevant? And that's if you look at Melanopsin knockout mice. So if you look at Melanopsin knockout mice, they show the same switch from preferring green to preferring red, but the switch occurs when the green light is much brighter. Okay? So they're much more likely to see the red light as brighter than the green if you take away Melanopsin. So the loss of Melanopsin changes estimations of brightness in favor of longer wavelengths. So if we go back to our V-Cry display, what we should be able to do then, what should happen is this image should appear dimmer than that image. That should appear brighter. And actually, I can't show you because we don't have a V-Cry display, but that is one's perception of that when you do that. Okay. So just very finally, for the last 5 minutes, I want to talk about the possibility, not that the whole scene could be rendered in Melanopsin low and Melanopsin high, but that elements of the scene could be rendered in Melanopsin low and Melanopsin high, and that might make parts of the scene appear brighter than others by manipulating the activity of Melanopsin. And that would only work if Melanopsin is capable of providing spatial information to the LGN. So, the question then is, can Melanopsin convey information about spatial patterns? We've done quite a lot of work on this. I just want to show you one very simple experiment which addresses that problem. Okay. So in this experiment, we're recording again in the LGN. And remember that for mice, this magenta looks Melanopsin low, and this cyan looks Melanopsin high. So what we then do is we present this stimulus. We put on a magenta background, and we present in a pseudo-random order these horizontal bars that appear in cyan. Okay? So what mouse Melanopsin sees then is this. Those bars appearing as increases in effective radiance for Melanopsin. What mouse rods and cones see is that, an invariant target. Okay? So what happens then to the activity of neurons in the LGN? So this is a single representative example, and what we're plotting here is the firing rate. The darker the color, the blacker each of these bars here is, the higher the firing rate was. And we apply the stimulus at time 0. And on this axis is the position of that horizontal bar on the elevation. So if you can imagine that this is what the mouse is seeing, and when the bar appears here, this neuron completely ignores it, whereas when the bar appears here, it's strongly excited. Okay. So what that tells us is that units in the DLGN can use Melanopsin to monitor brightness in particular locations in visual space. The receptive field sizes are equivalent to those for rod and cone input. I haven't shown you that data, but it's very strongly the case. But you can see that the time domain is quite different, so the spatiotemporal resolution as a whole will be much lower than for rods and cones. Okay. So, iPRGCs constitute a small fraction of the ganglion cell population. They're directly photosensitive, thanks to their expression of melanopsin. They entrain the clock and also drive many other reflex responses to bright light. So in a mouse, they may be 700, 800 per eye, but they have a huge impact on the biology of the animal. They're maximally sensitive to 480 nanometer light, which is cyan. And the fact that they're there and active during the day and have a different spectral sensitivity to cones challenges established methods of lighting design. So for architectural and artificial lighting, we can think about how we can generate light of different spectral composition, perhaps for different times of day. But because melanopsin also contributes to perceptual vision, we can think about redesigning visual displays, perhaps by including an additional color plane. We can target it to melanopsin, we can augment the appearance of visual displays. And that's something we're working on at present. Okay, I need to finish by thanking the organizations who provide funding for this work, for which we're very grateful, and more importantly, the people who have contributed to it. So I can't pick out individuals from my lab because so many people have helped us. We're very grateful for our collaborators around the world who have contributed resources, particularly mouse lines, but also ideas and expertise. And with that, I'll say thank you very much for your attention.